So happy morning, one and all. Today I'm here to present ROS-based nanomaterials for cancer therapy. As we all know, when it comes to cancer, ROS plays a dual role both in causing cancer and also in the treatment of cancer, based on the several signaling pathways and also the oxidative stress related to this. Cancer is one of the deadly disease after comparing to the cardiovascular disease. Despite the available treatment methodologies like radiation therapy and photodynamic therapy, cancer has started to show ineffectiveness in the traditional approaches for these techniques, and now nanotechnology is now being a wide new approach for the treatment of cancer and is now being in the limelight of research in terms of medical research. Speaking about the nanoparticles, cerium oxide nanoparticle is one of the commonly potential nanoparticle, as it acts both as an antioxidant and also pro-oxidant. This activity is completely based on the pH of the microenvironment. So, the cerium oxide nanoparticles functionality is completely based on the valency of these cerium ions. In a normal cell, the cerium oxide Ce4 plus is reduced to Ce3 plus, thereby oxidizing the superoxide into a molecular oxygen. In case of a cancer cell, which is a highly acidic environment, the Ce3 plus is oxidized to Ce4 plus, thereby reducing the superoxide to H2O2. The further reduction of this H2O2 is also catalyzed by the catalase activity of cerium oxide nanoparticles, where it will lead to the apoptosis of the cell by various mechanisms like mitochondrial disruption, protein oxidation, and also denaturation of the DNA. So, this is the mechanism in which cerium oxide nanoparticle is implicated by targeting cancer cell. And also, when in this study, when they treated albino mice, fibrosarcoma having albino mice, was treated with nanoceria, you can see that a drastic decrease in the tumor volume in case of a treated cell compared to the non-treated cell. So, also they estimated in this image also you can clearly see this is the tumor in a fibrosarcoma having diseased mouse, and this is the absence of the tumor or the reduced volume of the tumor. Also, the biodistribution studies tell that it is highly distributed in the tumor, whereas certain distribution is also seen in the liver and also in the spleen. From the study, they have estimated that a very small amount of quantity of this drug, that is 0.5 mg per kilogram, is enough to attenuate the tumor growth. Despite that, high concentration also did not cause remarkable side effects. So, also the mechanism in which it caused the apoptosis can also be evaluated by these two expressions. BARX2 pro, which is a pro-apoptotic gene, was increased. So, and also the decrease in the anti-apoptotic gene was observed, where we can tell that this increased the chances of apoptosis by blocking all the anti-apoptotic genes. Also, in this particular thing, the antioxidant activity was studied, where what they did is that in a particular cell, they gave an oxidative stress with the H2O2. The cells with cerium oxide, treated with cerium oxide nanoparticle is found to show high viability, whereas cells without nanoparticles is the viability is very less. So, from this we can estimate that it has an antioxidant potential also in a normal cell. This particular nanoparticle was treated to overcome the limitation of CDT and PDT, where photodynamic therapy is based on the principle that oxygen is converted to the reactive oxygen species based on the photosensitizers. But in a tumor environment, it is a hypoxic condition where oxygen is not available. So, in that case, it becomes a limitation. So, in order to overcome that, and also another disadvantage was that the antioxidant that is present in the cancer cell would inhibit the reactive oxygen species that was present that is produced by PDT. So, in order to overcome all these limitations, this particular nanoparticle was designed. Like it has copper ions, ICG, which is a photosensitizer, and also tirapesamin. Tirapesamin is a hypoxia activated anti-cancer pro-drug, which is activated into a highly toxic TPZ nanoparticles only in the hypoxic condition, which kills cancer cells. Also, the targeting of this particular ICG was used as a photosensitizer to oxidize our nanoparticle, and the targeting is based on the enhanced permeability and retention effect. the role of copper in this is that it acts as an antioxidant inhibitor, and also it catalyzes the Fenton reaction where H2O2 is converted into hydroxyl radical. Similarly, the hydroxyl radical by various mechanism, it will induce the apoptosis of cancer cell. As you can clearly see, the size of the tumor when compared to an untreated cell and with this particular nanoparticle irradiated with laser photosensitization. This laser is for photosensitization. So, the decrease in the tumor volume is clearly observed. You can see here also the tumor weight of in an untreated cell, and also with the laser induced cell, like a nanoparticle treated cell. And also this amorphous Fe nanoparticle, this nanoparticle was discovered to overcome the iron carrier nanoparticle, where the biooxidation of iron carriers was like occurring before the target itself. So, in order to overcome that, to specify the target, this particular nanoparticle was designed, where the nanoparticle was, this is the iron nanoparticle, iron molecule that is present embedded within the Fe2 plus fluoronic 127 and polyvinyl phosphate polymer, so that it will, this polymer will enhance the slow release of the drug, so that the bioavailability of the drug is enhanced. Also, the mechanism in which it is causing apoptosis is that Fenton reaction. So, it will the cancer cell that is highly with hydroxyl radical will be reduced to hydrogen peroxide will be reduced to hydroxyl radical, where it will induce the apoptosis. And also, based on the results, you can clearly see that the amorphous Fe nanoparticles treated with magnetization in the intratumoral region was found to like show very low amount of H2O2. In this study, they have estimated the hydrogen peroxide in the tumor cell, because the conversion of H2O2 into hydroxyl ion is the thing. If this nanoparticle will convert the H2O2 into hydroxyl radical, then it is found to be more efficient, right? So, in this study, intratumoral approach was like more efficient than compared to the intravenal thing. So, from this we can estimate that this is not a very potential target. So, we cannot administer every drug to an intratumoral, no. So, obviously it should show high things in intravenal. So, this is also like one disadvantage. So, obviously this particular nanoparticle, in this they use titanium oxide. Titanium oxide will be oxidized to titanium hydroxide, where it induces ROS based apoptosis in cancer cell. Also, to enhance the treatment, they have used a drug called cantharidin. Cantharidin is like a traditional Chinese medicine, which itself will cause effective which will effectively treat the malignant tumors. Also, in order to enhance the targeting efficiency of our drug, what they studied is that cancer cells have the expression of EphA2 receptor in cancer cell is high. This EphA2 receptor is nothing but a tyrosine kinase receptor. So, they identify targeting these receptor will be like a more efficient target of our particular drug. So, YSA peptide is like a ligand that binds to a receptor. It was chemically studied, and they synthesized this YSA peptide artificially, and to infuse that in the nanoparticle, they assisted PEG, polyethylene glycol, to coat it over the surface of the nanoparticle. And this targeting is also based on the enhanced permeability and retention approach. X-ray irradiation was provided for the photosensitization of the nanoparticle for oxidation purpose. And since this drug showed they have demonstrated the targeting efficiency also, it is very highly specific to cancer cell. Also, in order to test the cytotoxicity of the particular nanoparticle in both normal cell and cancer cell was depicted. When it was treated to cancer cell line, it showed like decreasing cell viability at higher concentration. When it was treated to a normal cell line like MCF10A, it did not show any cell cytotoxicity, where the cell viability is like so high. So, from this it can be told that it is very highly specific to nanoparticles, and it also kills the nanoparticles in a more efficient way at higher concentration. For that purpose, the apoptotic study was made, where the apoptosis of this particular nanoparticle was found to be high than compared to the blank cells without any nanoparticle. So in that approach, the final conclusion what I'm telling is like a ligand based nanoparticle will be a more specific target. See, for in my concern, serum oxide nanoparticle, which is even if has an off target activity, it did not show any potential side effect or any death in case of cells. So I would tell coating this serum oxide nanoparticle with a ligand of interest will enhance a very more prominent drug against cancer treatment. Thank you. And this is the reference. Yeah. Mm. Mm. What makes the difference? See, actually, the thing is, the reaction that is occurring makes the difference. For a normal cell, there will be more of H2O2 or some reactive oxygen species that is present. It will degrade the reactive oxygen species. In case of cancer cell, it will produce hydroxyl ions, which will induce apoptosis. So that is the difference. This is not a targeted one, obviously. But the thing is, it is from the experimental studies, it is found that it is more targeted to the tumor cell compared to the normal region due to the acidic nature of the thing. It is based on the characteristic of the nanoparticle. From this biodistribution study, they have estimated that it is highly targeting to a tumor compared to the normal tissues. I can't tell there is no toxicity at all. But from studies, it is estimated there is no clear effect. See, the cytotoxicity of this was assayed, and even in this particular study, they have administered it to the mice. The mice, they have extracted all the to metabolism. They have tested the metabolism. In that case, they have tested the creatinine level, glucose, and certain levels that is maintained. When it is compared with the normal and also treated nanoseria treated muscle, it did not show any significant difference. It was like similar. All the levels of the biomolecules were similar. So from that, we can estimate there is no harsh side effects. There may be side effects, which is not precise. Obviously, they'll be metabolized. They'll be excreted via kidney oil. They won't be like, they'll degrade, no. Everything is ions based and drugs based. Normal drug elimination mechanism. Yeah. Okay. Hydroxyl radical induces apoptosis in many ways. Like one particular thing is that it will activate caspase 9 protein. Caspase 9 is essential for inducing apoptosis. And the other thing is the BCL2 expression is like it will increase the expression of BCL2. BCL2 is a pro-apoptotic protein. So it will enhance the apoptosis in that way. So far as I was concerned, it is general statement that pH for cancer cell is acidic. Mm. Yeah, that is the thing actually, because so I don't get what you're finally asking. Mm. Mm. Yeah. Yeah. Obviously.
create this kind of either it should be or the production of superoxide should be more. Production of free radicals should be one of these. Should be the free radicals is more than as a general bacterium will be actually that again accept because we have enough backup to say that under any special condition the gas works. That is true. But the activity of antioxidant mechanisms depending on what if it is enzyme based. We don't really know the pH variation of the activity of each one is different. Then it might be production of the free radicals. I think everything plays a role, but apportion it to one, I'm not sure. I guess I'm sure it plays a role. According to one. The oxidative production of oxidative potential of superoxide ion is more. Superoxide anion, oxidative potential superoxide anion. Take a look at the table of reduction. It is context based. Anything below will accept electron from the there you'll find superoxide somewhere. Yeah, it is certainly potent. It is, but people like it because it's such a nice balance between high and low, and therefore they can make a lot of signaling and things like that. One of the reasons why we find hydroxyl is very necessary. But what you say is true, but one another reaction looks super radical. It could. It could. I mean, that's one of the things. Good. Yeah, that's
Yes, sir.
How these drugs are administered into the patient? Intravenously or
Obviously, it should be intravenously. In case of mice, it was given intraperitoneally. No, into patients, I don't know. Like, it is.
the serum based drug, right?
Huh.
Is off but if you can directly give it to the tumor tissue,
Hmm.
then it would be like easy to effective.
But you know, like, how will you find out where are the possible tumor
There
regions?
are techniques, right, for that.
Okay, but
Causing that patch.
if it is given intratumorally also,
There are techniques to actually figure out the cancerous tissues, and you can give directly the intravenously.
intravenously.
Yeah.
See, intravenously you can give. It will go and directly target our cancer cells. You're asking about intratumorally, right? Sorry, what you're
But
asking?
then, even if you look at like which particular tissue you have the cancer, then
Okay.
you can give the injection to that particular tissue. At least that would be like little bit decreasing of the rate of this.
Obviously. Yeah,
So how
in
it is administered into normal cancer patients?
that one, no, these are all based on the animal studies only, not on the human application. Yeah,
But these drugs are actually for humans, right? So they should have been
obviously.
tested on humans also.
Oh. Let's discuss that sometime. Yeah, but how we
Not like testing. This is like if the drug is like tested successfully, then it has to be successful.
what
Yeah.
kind of things is not up to the clinical trials.
This is animal study related.
Aren't they in chemical
Sorry.
these little particle drugs?
Ah.
Aren't they in clinical trials?
No, I don't know. I'm not sure. About that. Yeah. Clinical trials. No, I'm not sure about that. Can you be more louder? I can't hear you. That is based on the target. Cerium oxide nanoparticle will produce only in the cancer cell. But in case of titanium oxide, when it is oxidized itself, it will give reactive oxygen species. To ensure the target only, we are treating it with YSA peptide and all. So that it will go and bind. Only after binding, it will release the drugs. Because it is coated with polyethylene glycol, no? The drug release will also be like very slow. After that only if it binds to the target cancer cell, it will release titanium oxide. There will oxidize, will be like oxide. Also, there is a possibility it is acidic, so that the oxidation will also be like more enhanced. So that is also could be reason. Huh. Hmm. For that only they have in this case they have coated with polyethylene glycol. The surface coated with polyethylene glycol, it will prevent the leakage of the drugs. So only that is the most possible way. Also, it enhances the bioavailability of the drug. So that's this is actually a redox reaction. Cerium is reduced, therefore superoxide is oxidized. It will gain. No, it is not present in the physiological system. Cerium. In cerium. Yeah. Superoxide dismutase generally does this role. No? This antioxidant, it will convert superoxide to oxygen.
Okay, but superoxide won't get converted to oxygen or won't get scavenged in this mechanism by any other compound, right? Only cerium is creating this.
Yeah.
So far.
Okay.
So far, only cerium is creating this reaction.
Sorry, I don't get it.
In terms of metal, that is very rapid. Superoxide radical torque. You get scavenged by oxidation. You get free radical is getting scavenged by oxidation. That is very rare. So I asked if there is this question is other metals, any metals or any enzymes, because
Enzyme superoxide dismutase. Yeah. Okay, any other metal? No, I don't know. Okay, sure.
that will make targeting intravenous, put it into the vein, hoping that it will reach. And some of these drugs seem to act better on cancer cells compared to normal cells. Kill cancer cells better than normal cells because of the conditions in the cancer environment are exactly. That's the way they work. And mostly they are reactive oxygen. The reactive oxygen levels are high and therefore induce more the potential to go into dangerous level. In terms of injecting it, visualization without expensive, even with expensive you have that on time. Even that
that each and some of those now, I have come across detection of an anti-cancer drug to the region of cancer, probably because they don't know where. Right. We can detect the cancer cells. Yeah. Detecting the cancer cells is a problem, and access to the cancer cells, especially inside and so on. Cannot be easily reached, covered by organs, or not and so on. Those are things.
Actually, there are techniques available too.
I know, but you're okay. Hey, Christian. And yeah. Don't worry about it. Definitely not. Oh, you won't ask. See, when it is, I was just about to say after the presentation that you don't have to worry. Take rest, get well, and then we take care. EPL holds and evaluate. That's what I wanted to say. Let's, but don't go. Your health is a lot more important than a class. So go home and go to the hospital. Yeah, you're here. Yeah, good. Yes,